know it's crazy, but it's
You're watching Channel Zed, the world's leading zombie apocalypse channel, broadcasting live from Studio Zed.
makes me act the way I do.
And hello, welcome to another episode of the Dr. Zed Show on Channel Zed. We're going to have a discussion about evolutionary medicine and the apocalypse. As always, I am your host, Dr. Zed, aka Joe Alcock, MD. I'm a professor of emergency medicine at the University of New Mexico, ER doctor by day and night, also a researcher in evolutionary medicine. Today, I'm super excited. We're going to be talking about contagious cancer. We have some fantastic guests and co-hosts. We have Michael Metzger. We'll bring him on in just a little bit. He's a cancer biologist, evolutionary biologist who studies this topic. And we also, of course, have Athena Actipis. She literally wrote the book on the evolution of cancer. Hi, Athena. All right. So let's just jump right into it. Let's bring on Michael Metzger. I'll give a brief introduction as we bring him in. He is at the Pacific Northwest Research Institute. He more or less discovered a really remarkable phenomenon about this idea of clams possibly acquiring cancer by almost like an infection. So this is some new stuff. So let's bring him into the conversation. Let's see how this goes. There he is. Hi,
Hello.
Michael.
Hi, thanks for having me.
Absolutely. Michael, why don't you just briefly tell us about yourself?
So I'm Michael Metzger. I'm at the Pacific Northwest Research Institute. It's an independent research institute in Seattle. And I'm working on, like you said, the transmissible cancers in clams. And we're working on a number of different species here. This was a phenomenon that was actually first found in soft-shell clams in New England, but we found it actually in a number of species all over the world since then.
Truly remarkable discovery that's new as far as I know, having talked to you a little bit briefly before the program. But this idea of a contagious cancer. Now, I have heard about Tasmanian devils. Is that kind of the same thing?
Yes, yeah, that's the same thing. So there's only a few cases in the world of transmissible cancers like this. So just to start off, normal cancer that we think of is not contagious at all. So we know of diseases like HPV and other things where there's infectious agents, viruses that can cause cancer in people. So with HPV, there's a virus that jumps from person to person and causes cancer.
HPV, that's the human papillomavirus?
Yes, exactly.
So I'm thinking about cervical cancer.
Right.
Which is why we now have a vaccine called Gardasil that we can actually prevent cancer, which is a remarkable achievement in and of itself. But you're saying so the Tasmanian thing's different.
This is Tasmanian devils and clams. This is all different. So in that case, the virus moves, but your cancer cells themselves stay within each individual person. In the Tasmanian devils and dogs and in bivalves. So those are the three types of transmissible cancers that we know of. The cells themselves actually jump from one animal to the next and spread. So
it's not just a virus getting in and causing the cells to change. We're talking about the actual cancer cell going from one member of the species to another member of the species. But if you clone the cancer, it's all the same thing, right?
Right. So it's the same cancer. And that's exactly how we really identify it and diagnose it in the first place, is that you can see genetically that the cancer in one animal did not come from that animal. It came from a completely different individual from a different time.
All right. So, but Pam Winfrey here is asking this question: is it the virus that allows for the transfer of cancer? No, that's a good question, right?
Yeah.
Tell us about that.
In all of these cases so far, we actually don't know of any viral involvement in these. So it's actually just the whole cancer cell. So it's different from the human papillomavirus situation. So we don't know what caused the original founder clam or founder devil to have cancer, but then we know that those whole cells have jumped and basically engraft. Similar when you think of a metastasis in a single person, you have cells from a primary tumor site that can go and engraft into a different part in the same host. This is basically a similar sort of thing, but instead of just within a different host, it's going from a completely different individual over and over again and spreading through the environment.
Okay. Now, I mean, Tasmanian devils are kind of going with this zombie theme. They really are transformed by this contagious cancer. And part of their biology really facilitates the transmission of that contagious cancer, right? They're kind of aggressive, snarling, spitting beasts.
So the transmission of the Tasmanian devil facial tumor definitely builds on their own behavior, in which case, where they just sort of, by their nature, they bite each other quite a lot.
I've got a picture of that here.
There we go.
Thanks, Athena.
There's a nice snarling one.
There's a great picture of a little Tasmanian devil. So yeah, they develop these facial tumors that can be pretty disfiguring and awful.
I've got a picture of that too.
You got that?
All right.
Ouch.
Ooh, there we go.
Looks like a big erosion, a big ulcer on the face of underneath the ear of that poor little Tasmanian devil pup.
And this was a picture that was like not totally gross.
Yeah.
Just telling you, like there's some that are just so really just unbelievably gross. I didn't want to put a picture up.
It basically causes the death of the animal because they're no longer able to eat because it disfigures their face so much.
Yeah. So as Christina is pointing out, this is definitely a zombie apocalypse type situation. Certainly is for the poor Tasmanian devils. Of course, they are endangered, and this situation's not making their lives any easier, is it?
No, it's not. I think it's something on the order of 80% of Tasmanian devils have died due to this disease.
Okay. So we've talked about these Tasmanian devils. I think I saw some mention of dogs, and they have a venereal cancer, which is also kind of along the same lines.
Yeah, so the dogs have a single lineage of transmissible cancer that's called canine transmissible venereal tumor. And that's actually sexually transmitted, so another direct physical contact, but different sort of thing than the Tasmanian devil.
Right.
And so one of the unique differences between the Tasmanian devil and the dog cancer is that the Tasmanian devil cancer is actually very recent. It was first seen in 1997. So that's the first time anyone even saw these facial tumors. And they're pretty, like you said, they're pretty grotesque and pretty visually obvious if you see them. So that's thought to be quite new. And actually, since then, there's been two separate lineages of cancer developed within the dogs. And as Carlo is just pointing out, that in contrast to the Tasmanian devil one that's very new, the dog tumor is 6,000 years old. Basically, looking at the genotype of it, the original dog that had that primary founder tumor was 6,000 years ago, and it's been spreading as a sort of an asexual pathogen cancer since that time. And so we're talking about really huge timescales here, and where these cancers have been spreading.
Because I'm having a hard time wrapping my head around that. Clearly, we don't have any specimen in formalin of these 6,000-year-old cancers. How do we know the timing? How do we know that?
So that's done by a couple of groups looking at the genotypes of those cancers and sequencing the cancers we have now, and comparing that to cancers, or comparing that to normal dog sequences. So there's actually a huge amount of resources of genotypes of normal dogs of all sorts of breeds, and we have sequences for wolves and other related animals as well. And so if you look at the number of mutations that have occurred in the cancer relative to anything that looks like a normal original host, based on sort of making a molecular clock for how long those mutations would have taken to occur, the estimate is something, I believe the most recent paper is something on the order of 5,000 to 8,000 years ago. There's a bit of a window there because we don't know exactly how fast these things mutate. But that's been, I think, Liz Murchison's group in the UK has been one of the major leading people in that, and also Elaine Ostrander's group.
So we have a couple questions from the audience about, what happens over time? Do they evolve resistance? And in this case, the devils or dogs, what happens? Do they evolve mechanisms that help them to not become infected by the cancer?
Yeah, so with dogs, it's actually not as pathogenic as the Tasmanian devils. In some cases, it can actually regress, and it's actually relatively treatable with chemotherapy when it's found in pets and things. But in Tasmanian devils, it is a very serious issue since it's causing so much fatality within the animals. And for a long time, there were no cases of animals getting better. It was essentially 100% fatal. But in recent years, there's been thought that it is potentially there may be some resistance to it. There have been a handful of cases of animals that have cleared their cancers, and there's some really interesting work coming out of the analysis of those to try to figure out why those cancers are able to become sort of rejected by the hosts, and if there is evolution in the hosts. We're really interested in that in clams and other bivalves as well, because it looks like in clams and other bivalves, these bivalve transmissible neoplasias that we see, their outbreaks initially, sometimes in the order of 90% of the animals becoming diseased, and huge population die-offs. And so that's a really strong selective pressure for the evolution of resistance in the hosts. And so we're really interested in looking to identify how these clam populations are becoming resistant to these cancers. It looks like with the soft-shell clam cancer in New England in the 1980s, as well as Prince Edward Island in the early 2000s, there were huge population die-offs. But now when we go back to some of the same populations in New England, we see that prevalence is only about sort of 1 to 5%. And then we've seen a couple of, at least a handful of cases in the lab where we followed animals, and we can see that some of them will actually get cancer at a higher level, and then it will regress. And so we're just starting the analysis of trying to figure out how that's happening and how the animals are becoming more resistant to the disease.
Yeah, so when I was a kid clamming on Cape Cod, probably for a similar species, the steamers.
Yep, that's it.
That's the one, huh? We had to worry about die-offs periodically from red tides, but now we're saying that we have to worry about cancer too.
Well, this is a little bit different. So with red tide and some of those other pathogens, you don't actually want to eat the animals that have the red tide, and there's sort of paralytic toxins and things. You don't want to eat those. With soft-shell clams and the transmissible cancer, there's no risk to humans to eating these things. So we should mention that up front. If a clam has cancer and you eat it, you will not get the clam cancer.
Thank goodness.
What a relief.
I know.
So, Michael, I'm just curious to hear a little bit about how did you start studying these clam transmissible cancers?
So I started when I was a postdoc in Stephen Goff's lab at Columbia. So I was in a retrovirology lab, which is mostly human and mouse retroviruses, which is sort of an odd way to get into this. So these outbreaks, like I said, the outbreaks of these cancers had been known for a long time, and they'd been studied since the 70s and late 60s in some species, where we knew there were these big outbreaks of leukemia-like cancers in bivalves. And so people had thought that there was probably some infectious cause. And for a long time, it was thought that there was a retrovirus that was the cause. And so Carol Reinisch, who'd worked on these diseases as well, came to my boss at the time, Steve Goff, and said, can we find the retrovirus? And so this initially was a project to find a retrovirus. And I was working on that with another postdoc at the time, Gloria Arriagada, who was working with me together on this as well.
And a retrovirus is basically like a virus that's able to kind of get back into the genome and then sort of express genes from there, and that's how it makes trouble.
Yeah, so retrovirus, like HIV, is in the retrovirus family, and so retroviruses, because of the way they work, they're actually able to modify the DNA of the cells and integrate into the cells that they infect. And in a number of cases, retroviruses have been known to cause cancers. And so some of these are very well known in mice. So retroviruses that can spread through populations and induce cancers.
And that happens in people too.
So there's not a very they're not acute oncogenic retroviruses in people, but there are so retroviruses can cause cancer definitely at a low rate.
And there are viruses that
yes.
contribute to human cancers. So.
Yeah, there's a lot of infectious diseases that can cause cancer. So yeah, we mentioned HPV, then HIV can cause cancer in people who do have HIV infections.
Hepatitis.
Yes,
virus.
Hepatitis. There's actually quite a few that do induce a lot of other cancers. There's even bacteria like H. pylori can actually increase your rate of cancer because of sort of repeated inflammation and things like that as well.
Okay.
So yeah, there's a number of different infections that can induce cancer. But when we started working at this, it was just basically a side project to find a new virus in clams. And what we found out relatively quickly is that there was no virus that we could find at all. And we actually found a thing called a retrotransposon, which is related to a retrovirus, but I won't get too much into the weeds there. And what we ended up finding actually is that instead of being caused by a retrovirus, the other crazy hypothesis at the time was that it was actually a transmissible cancer. And that's what we ended up finding. Because whenever we started looking at genetic markers within the cancer cells themselves, we found out that all the different cancers that we're getting from all over Prince Edward Island and even Maine had a lot of the same genetic markers. So all of the cancers all along the East Coast were more similar to themselves than they were to even their own hosts. And that's when we found out it really had to be this transmissible cancer phenomenon.
So these are cell lines that really are kind of undead, right? They're kind of immortalized in a way, right?
Yeah. I mean, if anything really sort of deserves the definition of undead, it's you can't really call these cells, these cancers, sort of, they're not a live dog, they're not a live clam, and they haven't really been a live animal for decades to centuries to millennia. And yet they're still continuing to grow and infect and essentially sort of become a different life form, if you will, sort of more of an asexual parasite than their original host. So it's sort of hard to find a definition for them.
Wow, yeah. And so it's almost like you kind of go from having a multicellular species to having a mostly unicellular species.
Yeah, it really is. It's going from sort of multicellular organism to sort of its unicellular, asexually reproducing parasite species. And it is similar to cell cultures and things, human cells like HeLa cells that we have in the lab.
So I want to actually ask you, Athena, since you wrote a book on cancer. You should tell us a little bit about your book and tell us kind of what your view is on this. This is really kind of, these are cell lines that have sort of escaped their restraints and have hijacked their host in a way, right?
Yeah, well, I think that one of the interesting ways to sort of think about what transmissible cancer, contagious cancer is it's sort of like the next step after metastasis, if you will. So, you have, a tumor, it's in the body, it starts to invade, it starts to metastasize, go to other tissues. But if you have a population that is sufficiently genetically similar to itself, like the Tasmanian Devil population, and you have a mechanism for those cells to get into the new organism, then it's not that far from like what you would associate with metastasis in terms of the mechanisms. And I think when you do have populations that aren't very diverse or are immune compromised, or like in the case of the clams that Michael studies, there's this possibility, right, for the cells to actually just go in the water and then get sucked into the filters. So to me, I see it kind of as an extension almost of metastasis. And I think it's, one of the really, really important open questions, honestly. It's like, how much has contagious cancer shaped the evolution of cancer suppression mechanisms? It's not something that people really talk about, other than like me and Michael and Liz Murchison and a few other people, like when we get together at a conference. But it seems like, it's potentially a whole class of cancer suppression mechanisms evolved to limit the spread of transmissible cancers.
that is a fascinating way to look at it. I should mention that the book that Athena just wrote is called The Cheating Cell, and it's available on Amazon and other booksellers. I should single them out, and published by Princeton Press.
Yeah, thanks for the plug, Dr. Zed. I appreciate it.
You bet.
Anyway,
You
I'm
bet.
curious, Michael, what do you think in terms of like how much has contagious cancer potentially shaped the evolution of life and cancer suppression mechanisms?
Yeah, I think that's really quite possible that it has. I mean, as you say, cancer has really been a problem. As soon as you had the first multicellular organism, you have the potential for cancer to cheat and to become sort of a pathogen of its own host. And I think these transmissible cancers, while we are just identifying them, that doesn't mean that they're new. It just means that we're just seeing them. And particularly in bivalves, the more we look, the more we're finding. And I think it's quite likely that they've been affecting these animals for long periods of time, and we're just seeing the current wave of the infections. And things like, so some of the major barriers to having a transmissible cancer are things like the MHC response in vertebrates, the major histocompatibility complex. So the same thing that makes it so that if you want to take my kidney, you're going to have to take immunosuppressive drugs to keep it, because your body will reject it as foreign. That's the same mechanism that would keep you from getting a cancer from me as well. And so that blocks this. And so, right.
Unless I was an identical twin, and we shared those same immune markers.
Or if you're taking drugs to suppress your immune system after an organ transplant, right? I mean, we haven't really talked about transmissible cancer in humans, mostly because it's something that's extremely rare, but also because our immune systems usually take care of it, right? So the few examples that there are in situations where you have immune suppression for one reason or another, basically, right? So like organ transplants, it's possible. And there's cases, right, where someone did get a cancer as a result of the transplant. But if you look at like the benefits of getting the organ versus the risk of getting cancer, there's no question you should take the organ transplant. But it is something that it's possible in humans.
Pregnancy too, right? That's another situation in which you can get cancer from another human, right? A placenta, a sort of modified placenta, can become invasive.
That's the other case. There's been sort of organ transplantation as well as maternal to fetal transfer during pregnancy. There have been some very rare cases of that. Yeah. And in the cases of sort of mother to child transmission, you're not identical twins, but you've got sort of half the same genotype. And one of the really interesting things in one of the studies that looked specifically at the MHC loci, these are the genes that determine whether or not one cell is a match to another cell. When they looked at the cancer cells that grew in the infant, what they found is that it had lost the half that didn't match. So there has been a genetic mutation where it sort of deleted the copy that didn't match and only kept the copy that did match. And that sort of tells us that this self-non-self recognition in our cells is really, really powerful, and that it sort of had to throw out the half that didn't match in order for that cancer to continue to be pathogenic and to evade its host's immune response.
Interesting. Hey, Michael, we have a few questions or comments from the audience about cross-species transmission of cancer. Liz is saying, well, great that, we can't get cancer from other species, but are there cases of species transmitting cancers to other species?
There are in bivalves, but only between some pretty closely related species so far. So what we've seen is that the first cross-species transmission we found was in some bivalves in Galicia in Spain, and it was one type of clam that's called Polyditapes aureus, and it was actually, when we looked at the cancer within that animal, we found that not only it wasn't just a few SNPs different, it wasn't just different in a few places like a different individual, it was actually from a completely different species, and that was a different species of clam in the same family called Verrucous corrugata. So we can see a small number of cases. We also saw more recently, looking at mussels, there's a whole bunch of species in the Mytilus genus that are all over the world. And we found recently a cancer that arose from one species, Mytilus trocellus, and has now been found in two other independent species, Mytilus edulis in Europe and also Mytilus chilensis in South America. And so the same one has jumped to multiple different species. We don't see anything very large transmissions, so transmissions from like mussels to clams, that's not actually going to happen. That's actually been experimentally tried in the lab, and yeah, that never grows. But within closely related species, we do see it. And we actually don't know what that block would be. We don't know what keeps the mussel cancer out of the clams and vice versa.
Yeah, so Carlo here is saying we humans should watch out for Neanderthal transmissible cancers. So I guess maybe lucky or unlucky for us, we don't have a lot of very closely related hominin species that could transmit some kind of contagious cancer to us.
Michael,
what do you think about humans and contagious cancer? Or Athena, why don't you jump in?
Well, I since we only have a few minutes left, I wanted to ask Michael about the ecological impact of these cancers. You know, so what does this mean for clam species, and also, you know, how has human action affected the spread of these transmissible cancers?
I'm sure we have nothing to do with it, right? Just humans not involved, right?
No, not at all. Well, that's not 100% true. So likely these things have been affecting bivalves for a long period of time, but I think there's a lot that we're doing that is now affecting them. So I mentioned the mussel species where there's cancer found, the same lineage of cancer found in Europe and in South America. And there's really sort of no natural way we can explain that without human intervention. There are lots of Mytilus mussels in North America and the northern hemisphere and southern hemisphere, but there's nothing around the tropical region. And so we think the only way that they could have really gotten the cancer from one part to another would be through international shipping. And mussels are well known to sort of adhere to the hulls of boats and to get into ballast water. And so all you really need is an infected animal hijacking hitching a ride on a ship, and then that can transfer these cancers from place to place. And there was just recently a report by an independent group that found that same lineage of cancer in the Sea of Japan in Mytilus trocellus. So this is basically a single lineage of cancer that has been spread all the way around the world, northern hemisphere, southern hemisphere, and that is almost certainly with the help of humans.
So a new spin on an invasive species in both senses of the word.
Yeah.
How apocalyptic is this for the populations of clams and mussels, Michael?
We don't really know. I think the optimistic scenario is the one we've seen from softshell clams, where that's been studied sort of the same, what appears to be the same lineage for a long period of time, where you did see these 90% huge population die-offs, but then it looks like the species is recovering and potentially becoming resistant to these cancers and evolving resistance to them. And so I think that's the most optimistic thing. It's just they have to get through the period where there's a massive die-off and not become extinct. And we don't know, that may have happened a lot of times as well. We just don't see that.
Well, we have a final question here from KS asking, is there potential to discover cures and ways of, treating cancer as a result of studying these clams?
So I think in terms of treating the clams, in terms of treating the Tasmanian devils and dogs, there are definitely people working on ways to do that. In dogs, it is treatable with chemotherapy. With clams, we're, in a sense, not so much studying this to try to cure the clams themselves, but try to understand how these mechanisms have evolved that allows the clams to become resistant. And yeah, so as Carlos was saying, we really don't know what mechanism these clams are using to become resistant to cancer. And whatever that mechanism is, if we can find a new way that an animal has evolved resistance to cancer, I want to know what that is, and I think that there's the potential for translating that into something that can help human cancer.
Great lessons to be learned from really phenomenal work that you're doing, Michael.
Thanks.
What a joy it was to bring you onto this conversation. What an amazing topic! Perfect for this zombie themed show. So I really enjoyed having you here.
Yeah, thanks, Michael.
All right, thanks for having me. It's been fun to talk.
Yeah, so that wraps up another episode of the Dr. Zed Show, and thank you for joining us for this truly apocalyptic discussion. So yeah, join us. Keep tabs on the show. We're going to have some more content coming up. We'll be talking about endotoxin, ways in which our cells can be poisoned by bacteria. Fun stuff coming up. So we'll see you next time.
It's crazy, but it seems so logical. I can't deny that there is something supernatural with you.